Then if we look at the cytoplasm of the cell, when we are breaking the cell down, remember, keep in mind that we broke it down into the cell membrane, the cytoplasm, and the nucleus, that we should identify exactly what the cytoplasm is, and we always refer to it as being that jelly-like substance. But when it really comes down to it, the cytoplasm kind of has a lot more to it than just being a jelly-like substance. There's actually two very separate components to cytoplasm, that being the cytosol, which is that liquid component, and then a cytoskeleton. And skeleton should really kind of imply to you that structure, all right, support. And that structure and support is all these different fibers kind of creating a network, all right? And that's the reason for a jelly-like appearance, that you have all of this liquid, that cytosol in the cytoplasm, and throughout the liquid there are all these fibers networking and web-like, all kind of spreading in every direction, and that's almost kind of at a microscopic level causing a jelly-like appearance. In addition, we also take the organelles and we lump those organelles into being a component of the cytoplasm. So let's focus on those organelles. First of which is what we're going to talk about are the ribosomes. All right, and ribosomes are actually structurally composed of protein and RNA, which should really kind of make sense since this is the site of protein synthesis. This is where the RNA is going to come during protein synthesis in order to be translated to form chains of amino acids into proteins. You can find it on the rough endoplasmic reticulum, which in that case, when we're looking at the endoplasmic reticulum, sorry about the bad drawings, right, it's kind of embedded on the surface of the rough ER, and it's also going to be, ribosomes are going to be found free floating in that cytoplasm. The endoplasmic reticula, all right, these are these membrane-bound sacs and canals and vesicles, kind of this crazy like network transport system. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum, and we should know that rough has ribosomes. So the rough endoplasmic reticulum is studded with ribosomes, and those ribosomes are where that protein synthesis is going to occur. And rough endoplasmic reticulum really can be used for transporting proteins. So those proteins that are made by the ribosomes on the rough endoplasmic reticulum, those are going to be transported by the rough endoplasmic reticulum. Versus the smooth endoplasmic reticulum, which can transport, but a big role is that it's actually going to be responsible for making lipids. It can absorb fats, and it breaks down drugs, all right, those toxic substances. So we've kind of got three separate roles here for the smooth endoplasmic reticulum. Showing here the idea of the rough versus the smooth. You can see that the smooth kind of has more of like a tubular appearance, rough kind of being that weird membrane with all these studded ribosomes. The Golgi apparatus is going to be these flattened membranous sacs, right? A lot of times what you saw was me drawing them kind of like as this last year in biology class, and those are the cisternae. The role of the Golgi apparatus is to modify, package, and deliver the proteins from the rough endoplasmic reticulum. All right, it's kind of referred to, I typically call it the UPS, right, packaging and delivering. And it moves things by vesicles. And it'll move things within the cell and out of the cell. Within, it's called vesicle trafficking. Out, we should know that it's exocytosis. Again, we're going to cover this a bit more in depth later, but you guys have been introduced to exocytosis in biology. Packaging by the Golgi apparatus can be for immediate release, so the Golgi apparatus may package it and send it out to do its job immediately, or it may be stored in a vesicle until it's stimulated. And what I mean by stimulated, for example, hormones. Cells that produce hormones and are going to be secreting hormones aren't just going to be secreting them all the time in the same amount. All right, sometimes hormone secretion needs to be halted, slowed, and then there's a stimulation that more of that hormone is necessary. So keep in mind that the Golgi apparatus doesn't just package it and send it on its way. It may package it and hold it for a little while. All right, so here we have a picture. We're showing the nucleus up in this region. We've got a rough endoplasmic reticulum, and then we've got our Golgi apparatus packaging molecules. Here are our little molecules, the green, packaging molecules into a vesicle, and that vesicle is actually going to merge with the cell membrane in exocytosis in order to release those molecules. So those vesicles being membranous sacs to store substances, and they're going to be used to transport substances into and out of the cell. And we should know that into is that endocytosis, out is that exocytosis, and vesicles can be formed by the cell membrane, which would be an example during endocytosis, Golgi apparatus for exocytosis or for trafficking, and can also be formed by the smooth and rough endoplasmic reticulum, which would again be that trafficking, because then those vesicles are going to be sent somewhere else in order for the molecules to be modified or changed or sent to where their ultimate destination is. The mighty mitochondria. We should know that the mitochondria is really going to be about energy, right? It's often referred to as being the powerhouse of the cell. And that is because mitochondria is the site of cellular respiration. And cellular respiration, we should remember, is the process of getting energy from food. So energy released from nutrients, what we're really talking about is getting your energy out of your food. But some of the other details about the mitochondria is the fact that the mitochondria has two separate layers, an inner and an outer membrane. That inner membrane kind of folds in and on itself into cristae. But mitochondria also have a very unique feature. And this information should be relatively new to you. And this is the idea that mitochondria actually have their own specific DNA, right? Small amounts of mitochondrial DNA. And because of that small amount of DNA that they have, they're actually able to divide on their own. So the cell itself does not actually need to make more mitochondria when more mitochondria are needed. The mitochondria can replicate themselves and create more based on energy demands. So this is just kind of showing you a picture of that mitochondria and showing you, here's an example of that outer membrane versus the inner membrane, and seeing where those cristae form, see where they kind of fold in on themselves for that inner membrane.